So this would be our asynchronous class. So we're already on LP4 in terms of concepts, while the learning outcomes, we should be doing it on the board, but some learning outcomes, of course, I will be teaching it also in the asynchronous type so that we would not miss anything. So nucleic acids, LP4, that is the fourth type or kind or classification of your biochemical compounds, major classification of your biochemical compounds. So nucleic acids basically are the RNAs and the DNAs. So the animal, usually everything which has a eukaryotic cell would have DNA on it, but some simple type of cell has actually some modifications of RNA or an RNA only. Okay, so in terms of chemical compounds, what compose the genetic makeup or information about creating a new cell from a cell, because the animal kingdom is just made up of cells or anything which we say that there is a moving life or life which moves. Unlike the plants, they do have life, but they don't move around or have a brain, just like that. So anyway, in nucleic acid, as you can see from that main text that we are referring to, um, your nucleic acids has also primary, secondary, and tertiary structures. Unlike your proteins, it has up to a quaternary structure. So primary structure of nucleic acid is the order of bases, meaning part of your nucleic acid is the basis, the nitrogenous basis. So anyway, to paraphrase, because you have similar subjects with respect to this topic, you have you have four type of nitrogenous bases. So what order would it what order does that primary nucleic acid has is we term it the primary structure of that nucleic acid. And then secondary structure is the three-dimensional conformation of the backbone. So if you have already that sequence of a nitrogenous basis, so it only repeats. We say that that is the order basis because that is for a one particular unit. So if that unit is being repeated, so the secondary structure is the three-dimensional conformation of that backbone. We call that the backbone. And the three-dimensional conformation of that, whether it twists in a particular place because of this, and that, so we call that rearrangement in a three-dimensional perspective is what we call a secondary structure of that nucleic acid. And then the tertiary structure is specifically for the supercoiling of the molecule. We can say that some nucleic acids are coiled, supercoiled, in fact. So some are just plain, probably, or they transition from a supercoil to a non-coil. So we call that structure of that nucleic acid, wherein it behaves from its, let's say, a stable structure. We call those structures as a tertiary structures. So that's how we see or uh, categorize, or if we want to talk about the structure of a nucleic acid. And then you have, of course, the DNA and the RNA. I think this is common. The DNA stands for deoxyribonucleic acid, and your RNA is ribonucleic acid. So the only difference is DNA is more complicated than RNA because it talks about the deoxyribose, um, wherein it is also an advanced RNA actually. So RNA is more basic than the DNA. So the NNRNA appear in the secondary and tertiary structures. So it only talks about the secondary because you have to have that the three-dimensional uh, contortions and of course the supercoiling for you to classify, um, for you to talk about the DNA and the RNA. Okay, so that's what it says. So I only highlighted what is necessary or the important things. But of course, you can have bits of information as you read along, and that's what I expect. You are reading along with the book. I'm just guiding you if ever there's a particular terms that is really hard to grasp, or maybe I just can point you out what are the things that you are missing from, of course, from organic chemistry and maybe some other minor subjects before the biochemistry. More on the chemistry and part of for your, from your biology probably. Then you have ribosomes. It is the sites of a protein synthesis in all organism. So you have a protein. Basically, we are just made of proteins, the building blocks or what makes us, our cells, and cells are just make up, I mean, the building wherein you hold everything, you transact everything. I mean, your cell does its thing. It has structures, and we call that proteinous material. We have already discussed it in the protein LPs. I think that is LP2, and the site where those proteins are made. Okay, because we're just made of proteins, and our bodies is usually keep on making proteins like enzymes, hormones. Your body, your cell and your body, whatever part of your body is, they're just making proteinous material, because part of yourself or part of the animal cell always dies, and that is proteins. Meaning, if you talk only about the chemical side to it, we say that those dead cell are stop functioning, or it it no longer, it already reached its life, and it now will disintegrate. And when those disintegrates, of course, there should be a continuous process of replacing those genetic makeup of the cell. So ribosomes is the part where it is made. So every cell has a ribosomes on it. I'm not really sure, but I do think there are always ribosomes because it's where your protein is made. Of course, we have a lot of cells, hair cells. I mean, no, not hair cells, skin cells, eye cells, your brain cells, of course. So most of them, or maybe all of them, has ribosomes. I'm not really well versed with the biological contents, but I'm just reading through what this text says. Of course, my perspective is on the chemical part because I'm a chemical engineer. So just forgive me of my ignorance, probably. So that's one thing. And then you have terms. You have I highlighted there. Tobacco. Ribosomes. Another is a self-assembly of tobacco mosaic virus in which the nucleic acid strands winds through a cylinder of coat protein subunits. So I just highlighted it because that's one of the example how ribosomes should be understood. So some, like the tobacco music virus, so it's a virus. So meaning it's a different type of cell. Usually a virus is like a dead cell, or the whole of it is an aggregation of cell that is dead and becomes alive when it attaches to a host. Tobacco music virus in which the nucleic acid strand, meaning the genetic makeup, the nucleic acid that it contains, winds through a cylinder. So there is a cylinder wherein the genetic makeup just winds through it, cylinder of coat protein subunits. So you have protein, a coat protein subunits. So that cylinder is a coat protein subunits, and then there is the nucleic acid um, genetic makeup of the tobacco-music virus winds up to. So and then you have nucleotides. So nucleotides are purine or pyrimidine bases. So again, if you look at the organic makeup of purine and pyrimidine, so they are somewhat similar. So nucleotides are the purine or pyrimidine bases. So it's either purine or pyrimidine bonded to sugars. So sugars, we've already talked about sugars. Usually the sugars that we're talking about is the simpler ones, the ribose and the deoxyribose. We can see more of that the following pages, which in turn are bonded to phosphate groups. So nucleotides have a sugar to it. You have purine, pyrimidine, and then you have sugars, and then bonded to a phosphate group. Actually, that is the blocks we call for the um, units, subunits of your nucleic acid. So we even call the whole nucleic acid DNA RNA as a polynucleotides. Okay, so because it's made up of nucleotides, so the nucleotides composes of three things: purine or pyrimidine, and then you have sugars, bonded to sugars. So it has a sugar part to it, and then which also has a phosphate group. So we can see the structure to it on the coming part. See, no, see, you can see here it consists of three parts. Okay, that's what I've said. And then you have nucleic acid bases, pyrimidine bases, and purine bases. So from the nucleotides, you have one part there is what we call the nucleic acid bases. It is talking about the pyrimidine or the purine bases. So those three parts, we can classify again them, those two types of nucleic acid, meaning you can say that nucleotides are composed of nucleic acid bases. So that's talking about the purine and pyrimidine. Okay, so that's what it says here. And then what's the difference between pyrimidine basis and purine basis? Nitrogen containing aromatic compounds. So you have the nitrogen and then your aromatic compounds. You are talking about the benzene rings that contains a six-membered ring. Of course, that is a benzene. And then purine basis is a nitrogen containing aromatic compounds as the same that contains six-membered ring fused to a five-membered ring. So a six-membered ring fused to a five-membered ring. So side to side. So you have a six-membered ring and a five-membered ring. How about the other one? Six-membered ring only for the pyrimidine basis. For purine basis, that is six-membered ring fused to a five-membered ring. So that is purine basis. And both of those are we call the nucleic acid basis. Okay? So what you have there, pyrimidine basis, single ring aromatic compound. See? So you have example there, cytosine, thymine, and uracil. So usually this is the difference also with respect to DNA and RNA. Usually the DNA has these three. And the fourth one differentiates from DNA and RNA. So the pyrimidine basis, so meaning DNA has pyrimidine basis, so which is usually cytosine, thymine, and uracil commonly occur. Cytosine is found both in RNA and DNA, see, but uracil only occurs at RNA. In DNA, thymine is substituted for uracil. So uracil substituted, thymine is substituted for uracil. Thymine is also found to a small extent in some forms of RNA. So in RNA, you can say cytosine, thymine, uracil. Okay. So that's it says. The common purine basis double ring aromatic compounds are adenine and guanine. So that's the complete makeup of your DNA. You have adenine. Um, the common purine basis double ring aromatic compounds are adenine, guanine, both of which are found in RNA and in DNA. So adenine and guanine are both present. So they only differ at uracil and thymine. Okay, and uracil and thymine are considered as a pyrimidine basis. So you can see there are four, as we've said, there are uh, uh, what do you call this, uh, bases. So pyrimidine base for your DNA, while your purine bases are also four, they only differ at the uracil and thymine part. The thymine, meaning the DNA has cytosine, thymine, adenine, guanine, while your RNA has cytosine, uracil, adenine, and guanine. Adenine and guanine is a purine base, while your cytosine, thymine, and uracil are pyrimidine bases. So that's what only says. So unusual bases with slightly. So there are still other bases, but they are not common. So we will only be looking at these four bases. If ever you'll be looking at other nucleic acids or bases, so they are case-to-case basis, of course, because they are not found in all. But usually in all, DNA and RNA, all of these four are present. So they are unusual bases with slightly different structures. Actually, they are still the same, but they have a little bit different to their structures. Okay, so that's what it says. That are found principally but not exclusively in transfer RNA. And it only occurs in RNA and only on the transfer type of RNA because there are also different classes of RNA. So in transfer RNA, there is what we call a unusual basis. Maybe we only have to discuss them in particular cases. But if it is not discussed, surely we can neglect them by now because biochemistry is a long subject. We only need some generalized concept to it. Okay, so that you will know how to where to find probably if you needed a little bit of details on your biochemistry. In many cases, the base is modified by methylation. So that is a chemical reaction. That's why in your learning outcomes, I I I included the chemical reaction part because, as I've said, as you are strong with your organic chemistry, especially on the uh, uh, what they call this, the chemical reactions, of course, you can the more you can understand biochemistry. So another term is a nucleoside. So a nucleoside is a purine or pyrimidine base bonded to sugar. So again, for your nucleic base, it has three parts, but the two parts there, the pyrimidine bases and the sugar without the phosphate group is another term which is what we call the nucleoside. Okay, that's what it says. So as you can see from the nucleic acid bases, the nucleic acid has three parts again, but the nucleoside has two parts, meaning one of those being removed, so you can already classify them as a nucleoside. So that's what it says. So I've also highlighted the lacking a phosphate group that is for nucleoside. So I think you have there your pyrimidine and your pyridine bases. So you have for your pyrimidine, this is your pyrimidine actually, and then you can see the slight modification so that it will be under the pyrimidine or pyrimidine or what we call the um, pyrimidine basis, meaning it is somewhat taken from pyrimidine, and this is your purine. This one, your six-membered ring fused to a five-membered ring. So you have the adenine and guanine. See the difference? So this one is the classification. They're classified as pyrimidine purine because maybe that is the precursor. So as the body makes its genetic makeup, so it tries to change this one into cytosine, thymine, uracil. For RNA, that is uracil and cytosine. For DNA, that is thymine, adenine, and guanine. So I guess the video is already long. So may I cut it from here? Okay, thank you.